Do we have 2 labs due tomorrow?
Yes, you have to fix the Daphnia
Daphnia?
report, right? And macromolecules, yes. If you did like Daphnia before, like the report was fine, you don't have to redo it. But what I asked just to write it along with the results, right? Because many of you just send me results, that's it. But I asked to go over their points of report, like title, hypothesis, results, and then discussion. So I ask students to redo it. Okay.
Molecule one. Which one's that one?
Macromolecules, you have one page actually on the blackboard. There are directions what to do.
Okay.
So that's all you have to do.
Okay.
This is the one that is posted. This is the one you have to submit to me after answering the questions. Okay. Okay, so yes, Victoria.
I was just gonna say for the lab for tomorrow, we take that right on Blackboard.
Yeah, like you just download it from the Blackboard, answer the questions, and upload it back.
Okay, all right.
That's the macromolecules. Yeah. I should say, in my opinion, just a second, I'm looking for my pen.
Also, Professor, just one more question.
Yes.
For the Daphnia, there's some of them that you didn't upload a grade yet, right?
Not yet, because I'm waiting for reports.
Right. Okay. All right. Just making sure. Thank you.
I just wanted like all students to send it to me, and then I'm grading them all together.
Okay.
And usually I'm doing this like on Tuesday. Okay. So back to material. So this is chapter number 4, and this is our chapter about cells. And first of all, the whole study of cells became available due to invention of the microscope. Something that we're going to talk on Wednesday about the microscope. And the reason is that basically most of the cells are not visible to human eye, right? Without magnification. So I should say the whole field of cytology, which is study of cell, started 19th century, right? And then the cell theory was outlined. And one of the principles of the cell theory is cell is the smallest unit of life. It's the smallest unit of life, and it can be independent unit of life. It can basically perform all life functions, carry out metabolism and energy production pretty much on its own. And cells come from pre-existing cells, right? You can't actually put in a test tube a bunch of molecules, do some magic, and create a cell. No, cells come from pre-existing cells. And bottom line, all organisms are made of cells. Organism can be made of only one cell. Organism can be made of many cells. And this is, for example, mushroom, which is a fungus, right? Greenock, which is giraffe gazelle, and a head louse, all made of cells, right? So one more time, here is the cell theory, which is one of the most important theories in biology. All living organisms are made up of one or more cells. All cells arise from pre-existing cells. And this picture illustrates basically something that I was talking about. I said you can't see cells. Most of the cells you can't see without a microscope, without magnification. An exception are the eggs, okay? What is an egg, guys? Talk to me. What is an egg?
Isn't it like a shell? Like something that holds life? I don't know how to explain.
Mm,
Like, what is an egg, or what are like eggs?
like what is it? What are eggs? What are actually they for? Don't say for us to eat, please.
No, no.
Aren't eggs actually like living organisms inside? Like inside the egg, if we were to like not eat it, then um, an animal would be born from the egg. I don't know. Like
It's basically a living thing developing inside the egg. Something like
Yes.
that.
Who, who lays eggs, males or females?
Oh, both.
Females?
Both.
Yes.
He
Legs.
only egg.
Yes.
No, yo no
Egg-
tengo tanta cámara, es para que me escuchar.
Okay, so eggs are laid by females, right? And eggs are laid by fish, right? It's not exactly fish. Beluga is not really. It's a whale, right? Oh, no, it's a sturgeon. Sorry, it's a fish. So could be laid by fish. They could be laid by amphibians, right? Like frogs. They could be laid by reptiles, like turtles and snakes. They could be laid by birds. And this is a human egg cell, right? We don't really lay them. Females, mammals, what they do, they just produce them and release them in the time of ovulation. So these eggs could be fertilized, right? And the embryo will develop. But mammals develop embryos inside the body, right? Inside the uterus.
Water.
While some organisms, actually many organisms, don't do that, right? So fish, for example, female fish produces a bunch of eggs, and male fish, all has to do just to come over and fertilize them, right? Birds lay eggs, right? And if, for example, this is ostrich, which is a huge one, right? If male is around, this egg will be fertilized. If male is not around, this egg still gonna be laid, right? So why eggs are huge? Tell me.
Isn't it proportionate to the size of the animal?
There's something about that, but for example, mammals, like even an elephant egg is not that huge. If you talk about birds, yeah, maybe, but
What? No, elephants can't have eggs.
That's what I'm saying.
what? What,
Elephants
David?
can't have eggs. Elephants are mammals.
Like, okay,
Yo, can you wake
ostrich.
my sister up? Okay. I got a 70. I didn't do it.
Okay, so guys, one more time. If we talk about, for example, chicken egg, right? If you talk about chicken egg, right? So chicken egg is relatively large. And this, again, this is just one cell, keep in mind, made of only one cell. So the size pretty much corresponds to the idea that the chick has to develop inside the egg on its own, right? Mammals have placenta, and the developing embryo will be fed through placenta and receive all necessary nutrients. Birds, reptiles, fish, amphibians, right? They will be on their own. So all the necessary nutrients have to be supplied with the egg cell from the very beginning, right? Who can tell me how long does it take for the chick to hatch from an egg?
One day, I think.
Really? For the chick to hatch from an egg?
Or chick to hatch.
Yeah, I mean, to develop,
21 days.
not to hatch. Yeah, sorry,
21
develop.
Oh, to develop.
That's 21 days.
To fully be born.
21 days, yes. So,
Oh.
yeah. So again, if egg is fertilized, by the way, eggs that you buy in the grocery store, can you get a chick from them? Chicks out of them?
No.
No. Right. Why? Why wouldn't you get chicks from them?
Because
I mean, don't they have like a farm or like a?
They're not fertilized eggs.
Because they're in
Exactly.
cold environments.
No, they're not fertilized, right?
Oh.
Again, female will ovulate every month, regardless if her egg cell is fertilized or not, right? The same goes for birds. They're still gonna lay eggs. And it doesn't matter if this egg is fertilized by a rooster or by male bird or whatever. But again, the reason why the egg cells are visible, why they're huge, because again, they provide nutrients for the development, right? Development that takes place outside the body. And some of them, they take care of the eggs, right? So chicken, like chicken eggs, the hen gonna come over and she's gonna sit on them, keep them warm. She's gonna move them around to make sure that they get oxygen, right? The oxygen is able to penetrate through the shell. Some just lay eggs and disappear, right? So they're pretty much on their own. And as I said, mammals, they made a step forward, right? They have internal development. Now, internal development is definitely limited, which means that not that many animals produce like huge amount of offspring in a litter, right? But if you take again, amphibians or fish, they definitely lay far, far, far more eggs, right? Comparing to mammals. Okay, anyways, important point, eggs are made of only one cell. And this is again, exception from the rule that you need a microscope to see them. No, you don't really need a microscope. And a human egg cell is the largest cell produced by the human body. If you take like a pencil and you touch the tip of the pencil, you touch paper with the tip, that's how big female egg cell or ovum. Okay, now once we talk about cells, cells in this world are divided into groups, large groups or categories. There are cells called eukaryotic and the cells called prokaryotic. So what is it about? Let me share the whiteboard. So karyo means nucleus. Now, it's not the same nucleus as you had in the atom. It's a different nucleus. It's a nucleus that is present inside the cell. So pro means basically before. So these cells are probably the first cells that appear during evolution, right? Prokaryotic cells are usually small, right? And prokaryotes are found under something called domains. Domains, we talked about them, right? We talked about them a while ago when we talked about taxonomy. So we talked about bacteria and we talked about archaea, right? So all prokaryotes are single-celled organisms. So they're all made of only one cell. Now, if you're made of only one cell, you don't really need much genetic material, right? So you have DNA, but it's relatively small because, again, it has to code for only one cell. So genetic material in prokaryotes is not organized into additional structure, right? DNA is not organized into nucleus. Nucleus, again, is like a central piece inside the cell. So it's not organized into nucleus because, again, it's really, really small, right? Let me go back. so prokaryotes don't have a nucleus, right? DNA is simply resides in the middle of the cell. So if you take a closer look, this is Nitrobacter. This is bacteria cell, right? And as I said, they're really, really small. They're much smaller than eukaryotic cells. So these cells have something called plasma membrane. That's basically all cells share, right? Around the plasma membrane, there is an additional structure called cell wall. You can find hair-like projections called pili. They usually hop this type of cells to touch the surface, right? You can find structure called flagellum. It's a tail that helps bacteria to move. Inside the cell, you can find the DNA, which is again not organized into separate structure. You can find interior filled with a jelly-like fluid called cytoplasm. And you can also see these dots, they're called ribosomes. So these are structures that are involved in protein synthesis. Remember, I told you the most important function of the cell is to make proteins. That's what we talked about on Wednesday. So these are the units that are involved in protein synthesis. So this is the prokaryotic cell. Again, a very simple cell. But I should say prokaryotes definitely more successful because their number is far greater in this world than eukaryotic cells. These organisms are majority in this world comparing to those who made of more advanced cell. Okay, so eukaryotic cells are different, and eukaryotes include plants, animals, fungi, and single-celled organisms called protozoa or protists. Okay, so eukaryotic cells can be also made of only one cell, or could be made of many cells. Now, if you compare them, and I said, this picture is not how to say, is not the same. Again, as I said, the cells are much, much smaller, far smaller than eukaryotic. So what do you see in the eukaryotic cell? You see also plasma membrane, right? The border between the cell and the environment. You see a nucleus, the central piece with DNA. You see cytoplasm that fills the interior of the cell, and you see small units, they called organelles. Organelles are like tiny organs that provide cell with its function, okay? So one more time, this is a very nice summary between eukaryotic cells and prokaryotic. Remember, prokaryotic bacteria, archaea. The rest are eukaryotic. This is more diverse, but this is more numerous, okay? No nucleus to enclose DNA, right? No organelles except for the ribosomes. They're much smaller. Eukaryotic DNA is inside the nucleus. Cytoplasm with organelles, and they're much larger than prokaryotes. Any questions at this point, guys? Talk to me.
Can I repeat what you said back to you, just so I can get it?
Like the comparison?
Yeah, okay. So a eukaryotic cell has a nucleus and other organelles inside of it, and a prokaryotic
Not in the nucleus. Okay, outside the nucleus, between the
outside
plasma.
the nucleus. Okay, I'm really sorry about that. Let me try to sum up again. I'm sorry for arriving late, Professor.
Let me summarize it. Just listen, okay? Let me summarize it.
Okay.
So, look, if you, again, unfortunately, you won't have a chance to use a microscope. But if you look at the cell under the microscope, right? This is eukaryotic cell. What you're gonna see? You see, first of all, plasma membrane. Again, membrane is structure that surrounds the cell and separates the cell from the environment. You see the nucleus. That's where genetic material is. Between nucleus and membrane, there is a jelly-like fluid called cytoplasm. And in the cytoplasm, you're going to see organelles, like small organs that provide their cell with a function. When you look at prokaryotic cells, you see their plasma membrane and there's something called cell wall. You won't see the nucleus, you won't see much of the organelles, you see the cytoplasm. In the cytoplasm, there is a DNA, and the only organelle that they share with eukaryotes called ribosomes. We're going to talk about them.
Okay.
Anything else, guys? Are you with me? Do you listen to me?
I am somewhat with you. I
Yes.
am actually with you. It's just
I'm listening.
You're
it's
listening?
just so hard to do it.
Okay. Guys, and listen, like, if there's something that,
I only got a 14 out of 20 on the test, and kind of
but as
been
I said, if you do their volcano video, you send it to me, you're gonna get.
I'm gonna be doing that tomorrow because
No, you have to send it to me by the end of today.
well, that's all right. I need baking soda, and what do I need for a volcano?
And vinegar and food coloring. If you
Baking
have
soda
and vinegar.
Baking soda and vinegar. Let me see if I got that.
Professor, I actually wanted to ask you something about that really quick. I did my volcano, but it didn't erupt. I tried it 3 times with the baking soda and the vinegar.
What did you put in?
I put baking soda and then I put vinegar, and I
It didn't erupt?
time, and it melted my entire volcano.
Wow.
Yeah, so I was gonna ask you if there's another way I can
what
do this.
What would I say? Put maybe like a bit more baking soda.
I did 3 times. I have no more clay. It melted my volcano. I took a video of the effect. I can
Send me this. Send me whatever you got. Send me whatever you got. I'll take a look at it. Okay. So,
This has been a very bad week for me.
Okay, so let's go back. This is something we're gonna talk about today, right? So this is animal cell. I'm gonna give you overview, and then we'll talk like, part after part, right? So animal cell and plant cells, they share a lot of structures together, but still there are some differences. And I like to compare cell to a city, right? So a city that is self-sufficient, okay? So a city that can actually provide on its own most of the basic functions, right? So that's why cell is a basic unit of life, because it's self-sufficient. As I said, if you look at the cell under the microscope, the first thing you're going to see is cell membrane. So this is the cell membrane. Cell membrane surrounds, protects the cell, and keeps cell's contents together. In Europe, you can see in some places, very old cities, they had city walls, okay? I'm sure you heard about Moscow. Yes. Yes, guys?
Yes, I have heard about Moscow and the city walls and Red Square and everything.
Red Square, yes. What's the name of the city wall?
Oh God.
That's where the Russian government sits, right? Kremlin, right? So Kremlin.
Oh God, I thought it was Duma. No, Duma is the parliament. Sorry.
That's the parliament. No, but Kremlin is actually city wall, right? It was built long time ago when Moscow was a relatively small city, and the function of the city wall again is to protect, but also city wall has gates, right? So the same goes for the cell membrane. It has gates. It has towers to check whatever comes in, whatever comes out.
So that's the rough endoplasmic
No, I'm talking about cell membrane.
cell
Plasma.
membrane, plasma membrane.
Plasma
Okay, finally found it. Sorry about
Okay.
that.
So, which means again, plasma membrane controls whatever comes into the cell, whatever leaves the cell. Central piece is nucleus, right? So this is the nucleus. And inside the nucleus, there's genetic material, right? So genetic material is the most precious part of the cell because it's actually in charge of cell structure and function. So it has additional security level, which is a double membrane. So this is a nuclear membrane, right? And there are openings called nuclear pores for certain molecules to move in and out. But again, not too many molecules have access to the nucleus because this is the most important part of the cell. We're going to talk later, but inside the nucleus, there's additional structure called nucleolus. Nucleolus means little nucleus. And this one is in charge of important parts of the ribosomes. So again, genetic material corresponds to what? Guys, you talk to me, please.
Genetic material corresponds to what's inside the cell and has to replicate.
No, remember, one gene means what?
One gene. Oh God, why don't I know any of this?
Okay, lab. We're on the same boat.
David, what is one gene about?
Huh?
What genes are for? I explained this on Wednesday.
Oh yes. Genes are for the innate traits of animals and plants, and when people and genes, and they're for reproduction and replication.
Yes, of course, but no. Listen, on the level of their cell, I explained to you, DNA has genetic information, right?
Yes.
And one gene is information how to make what?
Oh, how to make a reproductive copy of
No,
yourself.
no. How to make
DNA?
one.
Oh,
Okay,
sorry.
Sarah, can you answer the question?
One gene carries the information, right?
Carries information about what?
About the makeup of the cell.
But can you be more specific? That's what we did on Wednesday. If you listen to me, that's how it goes. Like, one gene is a code to build one molecule, and this molecule is called
DNA?
no, DNA is a gene. But remember, which molecules make up our body? Molecules that have structure and function. You just had a quiz on that. Which molecules provide structure and function?
Carbon.
Carbon.
Yeah. Not carbon. Carbon is not a molecule. Carbon is an atom.
Oxygen.
Neutron?
What?
Oxygen.
No.
Neutrons, atoms.
No,
Neutron?
I'm not talking about atom. No, molecules that starts as letter P.
Oh, was it protein?
Proteins.
Sorry.
That's what I tried to.
Oh.
That's what I talked about last week, right? So genetic material is actually there codes, right? It's a code to build proteins, right? One gene corresponds to one protein. And one more time, the most important function of cell is to build a protein, right? So you have information inside the nucleus, right? But proteins are built outside. So basically space between nucleus and the membrane is called cytosol. It's filled with a jelly-like fluid called cytoplasm, right? And within the cytoplasm, you find the organelles that provide different function, right? So information about proteins is here, but you see this part? It's called endoplasmic reticulum. This one that has dots is called rough endoplasmic reticulum. On the rough endoplasmic reticulum, you have organelles called ribosomes. Ribosomes could be found here, could be found in the cytoplasm, right? But they are protein-making machines, right? So the whole area here is dedicated to protein synthesis. As I said, this is the most important function of the cell. So rough endoplasmic reticulum you can compare to industrial zone. Industrial zone with machines that make proteins. Part of it's called smooth. Smooth doesn't have ribosomes, and it's involved in synthesis of other molecules like steroids and lipids. Once proteins are made, remember proteins have to acquire certain shape, from the chain to complex 3D, right? So they have to be processed. The processing happens here. This organelle is called Golgi apparatus. So like again, like UPS, pickup and shipment, right? So proteins processed here, and from Golgi apparatus, they can move either within the cell or outside the cell. Cells need energy. It's called mitochondria, right? This is a power plant. Cells need sanitation. This organelle is called lysosome, provides sanitation. Cells grow and divide. So you have organelle called centrosome. This is the organelle that will actually help cell to undergo division. Okay, so as I said, similar to city wall is probably like a city hall because it's important information here. Industrial zone, UPS, power plant, sanitation, and it's like a building contractor, something like that. Okay, so this is a basic structure of animal cell. Plant cell is very similar, surprisingly, right? We're not that different from plants. But plants have organelle called chloroplast, and this is the organelle that is in charge of photosynthesis. Plants have a big sac with water. It's called vacuole. Vacuole keeps the water for the plant cell. So if you, for example, you want to make a juice and you squeeze tomato, that's where the juice comes from the vacuole. And plants, yes?
I noticed that the plant cells walls are much harder.
Yes, yes, because animal cell has plasma membrane. Plant cell, like bacteria, has a cell wall. This is made of cellulose. Remember we talked about cellulose on Wednesday? Guys, are you with me? I don't even like see some of you.
I'm here.
Yeah, you follow?
Yes.
Steven, are you there? Oh, nice to see you. Good morning, Steven. Finally, you turned the camera.
So you said a vacuole is a water bag for the plant cell, right?
Yeah. Okay. Yes, that's where you get the juice from the vacuole. Okay, so and there is something called endosymbiosis. So endosymbiosis theory. And the thing is, we're gonna talk about this like in a few. Organelle like chloroplast in the plant cell, and pay attention, plants have both mitochondria and chloroplasts, right? Animal cell has mitochondria only. Let me just do new share. I'm gonna clear it. Okay. So endosymbiosis. What is endosymbiosis? Endo means inside. Anyone tell me what is symbiosis? What the word symbiosis means?
Yes, you sound really low.
Somebody what?
You sound really low.
Symbiosis is like mutual what?
Mutual gain. Mutual.
Yes, exactly. Mutual gain, right? So
no, God, I forgot
Yes,
endo.
when two organisms actually benefit from cooperation, right? So in endosymbiosis, the story goes that there was a cell that was almost eukaryotic, and at a certain point, it incorporated independent organism, right? And this organism stayed inside the cell, became part of the cell, and became very useful part of the cell because it provided energy. This is mitochondria. Okay. For the plant cell, it was both mitochondria was incorporated inside in chloroplast. So mitochondria is energy. Chloroplast is photosynthesis. And why it's actually so important to know that? What's really special about these two organelles is that they have their own genetic material. Because again, long ago, they were independent organisms, right? So they had their own DNA. So mitochondria inside your cells has its own DNA. Usually DNA inside the nucleus is called nDNA, nuclear DNA. DNA from mitochondria is called mDNA, mitochondrial DNA. Now, all your mDNA you get only from mother. Why do you get it only from mother? Because all cell contents you get from the mother. Remember I told you egg cell is huge, right? So this huge egg cell carries all cellular structures. What your father gives you, what your father gave you. What did you get from father? Catherine, can you say, please?
Professor, can you give me a minute? I'm trying to write everything down. You went really fast.
I'm fast? Okay, I can slow down, absolutely.
You said nDNA came from your mother only, right?
Yes, because I'm saying that all organelles come from mother.
Okay.
Egg cell carries genetic material in all cellular contents, right? Sperm cell carries only what?
Is it the instruction?
Sperm cell carries only DNA, right? DNA only. Mother gives DNA and all parts of the cell, right? So basically you get extra DNA from mother, right? So if this is an egg cell, right? So you get nuclear DNA, right? And you get DNA. So slightly more DNA you get from mother than from father. And as I said, you can get it only like maternal line. Now, what's significance of this DNA? Surprisingly, it's quite significant. And genes that mitochondria carries are responsible for energy production. If you talk about mitochondria, mitochondria gives you 95% of energy. And if there is a problem with this genetic material, child can suffer from a serious syndrome that will affect child's ability to produce energy. Now, there's something that I want to show you. Are you with me, guys?
Yeah.
Yes.
Yes.
Have you ever heard about babies born to 3 parents?
What? No, this is actually quite curious.
Don't they like get like a little thing and they mix everyone's DNA? No.
Okay. So first of all, what's the definition of parent in biology? Before I'm going to show you. Parent is who contributes genetic material. So in biology, not anthropology, not whatever, not psychology, just biology. Parent is the one who gives genes, right? And we usually say mother gives genes and father gives genes. What if third party gives genes? It's also considered parent.
Like a surrogate? No?
No, let me show you. Let me show you this. So baby born to 3 parents.
Ah, the nurse. Who would have thought?
My cute.
So I think this is a good one. Okay, so let me show you. Okay, imagine a woman, right? So she knows that she has mutation in her mitochondrial DNA, right? So she produces egg cell, right? And what you can see, this is like faulty mitochondrial DNA in her cell. Now, another woman donates her egg cell. So they leave the cell, but take out the nucleus and put the nucleus of the mother, right? So she's like also considered the mother, but a nuclear DNA definitely contributes more genes. So you see this manipulation? You take the healthy egg cell, take out the nucleus, and put the nucleus of a woman who has problems with her mitochondrial DNA. So she basically gets a cell that's called like reconstructed egg. So in this egg cell, there is maternal, like nDNA from one woman and mDNA from another one. If this fertilized by sperm, right? So this child gonna have 3 parents. Mother who gave nDNA, mother who gave mDNA, and the father. Is it clear?
So this isn't a surrogate?
No, this is not a surrogate. And you see this is, you see happy baby born to 3 parents. You see here.
Then the baby will have traits of all 3, or?
The thing is, listen, mitochondrial DNA is not that big comparing to nuclear. So I believe like 90%, that's definitely nDNA that matters, right? But what really matters that the child won't have a genetic disease associated with faulty mitochondrial DNA. That's the most important thing. Make sense? Guys, any questions? So it's not something like a threesome or whatever or surrogate. No, this is just pure genetic manipulation. I should say molecular biology manipulation, which is it's a quite advanced technique, I should say.
What would it have wrong with it with the faulty mitochondrial DNA?
As I said, genes pretty much involved in energy production. It's a serious disease, genetic disease, when a child actually, I just want to show you here, open it here. This is mitochondrial transfer, but this is pretty much, it's not explained here, but genes that control production of energy. And if you have mutation with them, your cells won't be able to produce enough energy to sustain basic life functions. So those babies without this procedure will die within a few years. Okay? Any questions here? Just 1 second. So symbiosis theory, one more time, it's about organelles like chloroplast and mitochondria that have their own genetic material, right? And it's not exactly known when did it happen, when they were incorporated, right? And just keep in mind, these two organelles, chloroplast for plants, mitochondria for plants and animals. And this basically shows endosymbiosis is incorporation of the whole entire organelle. This is called invagination. Invagination is basically acquiring genetic material from outside, taking it from the outside and bringing it in. But I don't think it's really relevant for us today. And what we're gonna start talking about, we're gonna talk about plasma membrane for another like 10 minutes and then you take a break, okay? Are you with me, guys?
Yes.
Yes.
Well, I got to admit, I'm beginning to think that my otherwise okay record in this class is going to start slipping. I mean, first it was chemistry, now we're going into real biology, and I don't really understand any of this.
We didn't even start lab. We didn't even start, okay? I just gave overview of the cell, right? I explained about two organelles that are independent. I mean, they were incorporated into eukaryotic cell. It's just the beginning.
Yes.
So please be patient. And you probably didn't hear that. I kind of like explained at the very beginning of the class that cell is probably the toughest part of the whole material. But once we pass cell, I promise you, things gonna be much, much easier. So much easier. This is probably, yes, as I said, it's a break point.
Well, I mean, I also didn't do so well in the last quiz, which was about nutrients. I also forgot the September 15th quiz. Was there a quiz on September 15th that we were supposed to do?
When was September 15th?
Never mind.
Last week?
Yeah, there was a quiz last week.
Okay, so guys, let's talk about cell membrane. And as I said, please be patient. You have questions, stop me, I will explain. You have questions, we talk more on Wednesday about the cell as well, okay? And I also don't think that like you are completely unaware of cell structure. You were exposed to this material during the school. So try just to bring your memories up. Okay, so cell membrane. If you go back to this picture, cell membrane is pretty much, it's a part of the cell that makes cell independent unit. Remember, cell is independent unit of life. Do you listen to me, guys? Do you, as I said, this material is hard, so you better listen today, just to make your life easier. Yes, William, I know you're with me. I know you listen to me. I see Ingrid, yes. Vittori, are you with me?
Yes, I'm sorry.
Okay, great. Okay, so cell is bordered by plasma membrane, right? It's a border that separates whatever inside the cell from whatever is outside the cell. Now, cells are usually, again, if you talk about single cell organisms, multicellular organisms, cells don't exist in dry environment. Cells exist, you know, in their fluids, right? So if this is a cell, this is called extracellular fluid. This is called intracellular. So both fluids have water, right? Now, if water will move freely inside the cell and outside the cell, what's going to happen?
I think what will happen is no, I don't know. I'm sorry.
It's okay. So
Together, right?
what?
It would run together, right?
It's not gonna make any sense. Like I should say, if water will move freely inside the cell and outside the cell won't be able to keep interior stable, right? So cell membrane should be able to repel water. Do you remember how you call molecules that don't mix with the water? What's the name for these molecules?
Non-soluble.
No. Hydro.
Oh.
Hydrophobic. What are hydrophobic molecules? Those that don't mix with the water. What kind of molecules are they?
Lipids?
Excellent. Yes.
That's cool. I got that right. Lipids.
I'm so happy. Yes. Lipids. Awesome. So cell membrane, yes, has lipids, and the function of the lipids is to repel water. So basically what happens, cell membrane is made of special molecules that are called phospholipids. Phospholipids, they have phosphate head, which is hydrophilic. This phosphate head is able to interact with the water, but they have tails made of 2 fatty acids that are hydrophobic. They repel water. And the organization is that, you see, heads face outside and heads face inside. And again, heads are hydrophilic, so they can interact with extracellular fluid, intracellular fluid. But the tails that face each other are hydrophobic. Water can't cross the cell, okay? Only molecules that are hydrophobic as well can cross the cell membrane. Any molecule that is hydrophilic can't. And remember, hydrophobic means they don't have a charge on them. Any molecule that has a charge can't go through this double layer, okay? So how molecules that are hydrophilic can move? They can move only through channels or carriers. So like remember gates? Remember I told you cell walls have gates? So these are the gates for the molecules that are hydrophilic. So I should say it's a very genius design, to my opinion, right? Because one more time, these hydrophobic fatty acids really keep interior of the cell stable. Any questions here at this moment? Dr. Meek?
No.
Yes, please. Yes, what's the question? Okay, now this structure of phospholipids, remember, double layer of these molecules is not rigid. It's very soft. And plasma membrane is actually, it's a very dynamic structure. If you think about, again, egg, the egg shell is not plasma membrane. What is plasma membrane in the egg? If you boil it, there's like a thin, whitish, like film, not film, like, you know what I'm talking about. When you peel the egg, there's like something like white layer that sticks to the shell. Yes?
Yeah.
That's the plasma membrane, okay? So within the plasma membrane, as I said, you have their phospholipids. You also have molecule of cholesterol. Cholesterol is the part of the cell membrane. And the function of cholesterol is actually to keep cell membrane stable whenever temperature changes. Okay, when temperature changes, when it's like getting colder, the molecules tend to stick to each other. When it's warmer, molecules tend to move apart. So cholesterol kind of like stabilizes the position of their phospholipids. within the membrane, you find a lot of proteins. Proteins play important function. Proteins can be receptors. Receptor means they're a sensor. This is chemical messenger that can bind to receptor and deliver message to the cell. Some proteins could be what's called recognition proteins. Recognition proteins in animals, in animals specifically, also in plants, they are part of the immune system. A lot of transport proteins, as I said, wherever is hydrophilic needs to be brought in or out. And the membrane proteins that can actually kind of like anchor membrane to certain structures, or they can conduct chemical reactions if they're enzymes. You can find carbohydrate chains that they actually also molecules that used for identification and for communication. Okay, so proteins can be receptors, can be used as recognition, transportation, and enzymes. And we can see faulty proteins can cause diseases. I think I talked about cystic fibrosis. I'm not sure if I talked to you. This is genetic disease. And in this genetic disease, cell membrane basically can't retain fluids, and these fluids are released outside. So a lot of extracellular fluid, and these fluids, for example, get accumulated in the airways, in urinary tract, in digestive tract. And if you have too much mucus, too much fluids, it's a breeding ground for germs. So in this genetic disease, they need a bunch of medications. They also need to kind of like unclog, to clear the airways. So you see this special vest that will help this girl to get rid of extra mucus from her airways. And again, this is one of the common genetic diseases, the cystic fibrosis, and it's pretty much due to abnormal function of proteins on the cell membrane. We can skip this part. And as I said, these are immune, like these proteins have immune function. And this, unfortunately, in some cells, like in T cells, which are part of the immune system, that's what the HIV virus can bind to. And that's how HIV virus ends up inside a cell. Okay, this is cellular HIV transmission diagram. You know, HIV stands for, guys, what HIV stands for?
Human immune deficiency virus infection.
Absolutely, awesome mood. Yes, it's a human immune deficiency virus. And you see, sometimes, you know, these molecules, again, our system, you know, can make mistakes, and instead of the right thing, let the wrong thing, the viruses, enter the cell, and that's how the viruses infect the cells. Okay, so 2 more things and you're gonna take a break, okay? So we're gonna talk right now, and after the break, we're gonna talk about their membrane transport. So you have a cell, and again, cell needs a bunch of things. Cell needs a lot of supplies. It needs water, it needs amino acid, it needs oxygen, it needs to get rid of carbon dioxide. So how things move in and out of cell. So generally there are 2 ways. Membrane transport, guys, that's gonna be on your next quiz, that's gonna be on your final, or sorry, midterm. Passive. Active. What's the difference between passive and active transport? Active needs energy. Energy comes from the molecule called ATP. So Active transport needs ATP. Passive transport doesn't need ATP. So passive includes diffusion, osmosis, facilitated diffusion, and also add filtration. Transport use pumps. In transport cold, we're going to talk about this. So cytosis and endocytosis. So one more time, no inner energy needed. Energy is needed. Now, before you're going to take a break, I want to hear from you. Do you need this? Do you need this?
Yeah. Two
Okay.
seconds.
I'll wait. You copy. I ask you a question, and you're going to take a 10 minutes break. Okay, by the way, whenever you see cyto means cell, exo means outside, endo means inside. Exo out, endo in. So my question to you basically, but I just want the whole picture. Can I switch? Can I just stop sharing? Okay, so my question to you.
Professor, this is gonna be being recorded, right? And you're gonna post it on Blackboard later.
It's recorded. Yes, it is. Okay. Now we actually I want whiteboard. Okay, so let's say you have a bunch of stuff right here. Whatever you want. It could be molecules, it can be people, right? And here you have one. So here you have many, right? Here you have little. If you refer to molecules, this is called concentration gradient. What is gradient? Gradient means difference in amount, okay? The difference is called a gradient. And again, gradient can be chemical, gradient can be in terms of charges. A lot of positive charges here and here you have a few, right? It doesn't matter, but as long as you have a difference, right? So imagine you have a room, right? Like people enter the room and accumulate here at the door, right? But the room is available for all of them. Where are they gonna move? Which way are they gonna move?
To the side, everywhere.
Everywhere. Spreading. So people move, molecules move as well, right? So when molecules move from high concentration to low, right? This is called diffusion. Diffusion is movement of substances from high concentration to low. Okay, so cell is not permeable to water, but cell is perfectly permeable to oxygen. So oxygen will enter the cell just because you have a lot of oxygen outside and a little bit inside. It's when you add food coloring, right, to water, it will spread even because of diffusion, right? When you add sugar to your tea, you mix it, that's diffusion, right? You open like perfume and you can smell, you know, a few feet away, that's also diffusion. Or we have like outside, not far from my house, we have Nabisco. It's a building where they make cookies. And sometimes you step outside and you smell like chocolate, smell, cookie smell, right? So it's another example of diffusion. This is one of the easiest, I should say. You go from high to low. And this is an example of food coloring. Okay, now there is something called facilitated diffusion. What is facilitated diffusion? Simple diffusion, you just cross the membrane, again, from high concentration to low. Facilitated diffusion happens for the same reason, but you need a door, you need an opening, okay? Oops, sorry, that's not what I needed. So diffusion, again, people enter the room and spread. Now imagine you have a room and you have doors and you have people outside. These doors are closed. The moment you open the door, people are gonna move in, right? So facilitated diffusion happens, again, you have accumulation in one place, right? But to balance, right? To move things, to have the same even concentration inside and outside, you have to open the doors, right? So water, remember I told you, water can't cross the membrane. Water needs a channel, it needs a door, right? So this door is open, water comes in. Doors are closed, the water can't come. Does it make sense? Does it make sense what is facilitated diffusion?
Yes.
You have to open the doors for the things to happen.
So facilitated diffusion is basically the process of opening and closing doors.
Yes, you open the door for, this is glucose, right? You open the door for the glucose.
Oh, yes. So through that illustration, I'm seeing facilitated diffusion is a type of opening, whereas simple diffusion is things passing through. Facilitated diffusion is like a special type of opening.
Exactly, exactly. So the next one is called osmosis. What is osmosis? Osmosis, wait, guys, let me just explain to you what is osmosis. So we have a few more things here. Osmosis is when you have a cell, right? And you have water inside the cell, remember, and you have water outside the cell. So certain molecules can move on their own, right? To make concentrations even, the water does the movement. So I'll go to the whiteboard one more time and just explain to you. So let's say you have a glass, right? And in between the glass, you put membrane, right? And this membrane, let's say sugar and but water. Yes. So you fill the glass with the water, right? And then you put sugar right here. So what's gonna happen? What's gonna happen? Again, I told you, sugar can't cross. You have here a lot and here you have none. But sugar is blocked. So to make concentrations even, the water gonna start moving through this membrane. And over time, what are you gonna get? Over time, here you're gonna have more water, and here you're gonna have less. This is osmosis. Osmosis is different from diffusion. Osmosis when water moves from high concentration of, from low concentration of substance to high until even. Osmosis is a super important way of moving fluids around the body. Most of the fluids in your body move through laws of osmosis, like production of sweat, production of saliva, production of urine. It's all interplay between substances and water in your body. I'm sure you heard that if you eat spicy food, right? If you eat something salty, your body tends to retain water. Have you heard about that? Have you heard about that? Tell me.
No.
Yes, I have heard about that. When you eat spicy food, you need to sweat in order to cool yourself down. That's why you retain
I
water.
thought I was the only one that happens to. I'm not gonna lie.
Of course, I would never know. I don't eat spicy food.
Okay, but listen, let me just give you an example. People get thirsty usually for two reasons. One type of thirst is just you are dehydrated, right? You're just dehydrated and you drink water. But another type of thirst is called osmotic thirst. What is osmotic thirst? You eat spicy. And right after eating spicy, what do? You drink water, right? You drink water,
Be
not
like something to
because
cool you down.
it's not to cool you down. No. What happens, you get too much
It's to
salt.
wash away the spices. It washes away.
Yes, it's not also washing away, but the thing is that in your blood, in your body fluids, you have certain concentration of sodium. This is salt, right? You eat spicy food, you get more sodium.
Oh.
You get more sodium, you need more water to dilute it. That's the whole thing. That's osmotic thirst. And yes, your body retains more water if you eat salty and spicy. So let me just give you, let me just show you here. This is isotonic and hypertonic, and basically there's something called tonicity. Tonicity is amount of fluids, solutes, inside the cell and outside the cell. Iso means equal, hypo means lower, hypertonic means higher. And I go back to whiteboard and I'll show you. Okay. So in your blood you have like a watery part of blood called plasma. I'm talking about blood. So you have plasma and you have the most common cell in your blood are red blood cells. So now, in plasma, concentration of the same salt, I'm talking about sodium chloride, is 0.9%. That's how much salt you have in your plasma. And this is how much salt you have inside the red blood cell. This is called isotonic. The concentration of salt inside the cell and outside the cell is equal. Now, let's say person lost blood, bleeding, right? And in this case, you need the person to give the IV fluids. Anybody knows what's the name of the IV fluid? You know, these bags that people get when they are in the hospital and they're dehydrated?
Like saline?
Saline, exactly. What is saline? Saline is 0.9% sodium chloride in water, right? That's saline. Can you give person just water as an IV? Regular, I mean distilled water.
No.
You're gonna kill the person. How are you gonna kill the person? Let me explain you how you kill the person. So this is 0.9%, right? And outside it's water, 0%. This is a solution called hypertonic. What's gonna happen next? Remember, osmosis. Water gonna move from low concentration of substance to high. So water gonna start moving into red blood cells. And red blood cell will swell and expand. So that's the reason why you shouldn't give IV just water.
I have a question, Professor.
Mm-hmm.
How much water would you have to drink to do that? Because people say sometimes you drink too much water.
Yes, the thing is that if you're gonna drink too much water, you're gonna start throwing up. Okay, it's not a good thing. So, for example, anyone here who's like, at least I don't know, like how's now, but used to work out a lot. Anyone here?
Once a week.
But if you work out a
Me.
lot and you sweat a lot, it's not recommended you to drink water.
Really? I took a whole thing of water after.
No, what is recommended to drink is actually electrolytes like Gatorade.
Gatorade.
Yes, another one is good is coconut water.
Wait.
Because if you just get water, that's what's gonna happen to you. Not definitely, it's not the same as you get at IV. But if you, for example, if you lost fluids, sweating, throwing up, don't drink just water. Drink electrolyte like Gatorade, like juice, like something, right? Okay, makes sense?
Have a lot of sodium in it.
What?
Gatorade and like Powerade, they have like a lot of sodium in them.
But that's what I explained to you. Exactly, that's osmosis. Because your cells will start swelling. You drink just water, it's not only red blood cells, all your cells gonna start swelling slightly because of the osmosis. Water gonna move into your cells, okay? Now, the last one. The last one, if you have a red blood cell, right? And you want to help a person, you want to make her IV, right, from scratch, but you messed up. And instead of 0.9, you make 9% sodium chloride. So concentration outside the red blood cell gonna be much higher. That's called hypertonic. So what's gonna happen to the cell? The water gonna move in or out? Tell me. In or out?
Out.
Out, exactly. Out. Gonna start go out. So the red blood cell gonna shrink and die. This is called crenation. Crenation is shrinking of red blood cell. Okay. So this is just example of osmosis, but one more time, the whole issue of retention of water, the whole issue of moving water from your blood to your tissues, from tissues back to blood, from your digestive tract into the blood, whatever, all this circulation around the body is due to laws of osmosis.
Okay. Okay.
You
Sorry.
have salt. Whenever you move salt, the water will follow. Okay? So if I go back here, so you have explanation of tonicity, isotonic, hypertonic, hypertonic. You have example here of red blood cell, right? And this is also about the, pretty much it's also about the plants, okay? And for example, you see in plants, if concentration of water goes down because of their dehydration, right? Water evaporates from the plant the same way like evaporates with our sweat. So you put it into the water and the water gonna move up, right? That's how you have a dry flower, like a not that flower, but you put it in the water, osmosis gonna bring water inside the plant. Okay? So whenever you have salt, one place, salts, sugar, whatever, it attracts water. It pulls water by osmosis, through osmosis. That's how plants also get water from the ground, right? The same thing. Or you know, the last example, osmosis, and I'm gonna move to something else. Last example, you know those toys that I remember, it's like an egg, right? And you get like a dinosaur and you put it in the water and it swells, grows, yeah?
Like a little sponge thing?
Yeah, something like that. This is osmosis, pretty much. Okay. And the last thing is active transport here. So in active transport, cells have to use energy. Why cells have to use energy? Energy has to be used when molecules have to be moved against concentration gradient. So diffusion, remember, is easy. Facilitated diffusion is easy. You have a lot and you have a little, right? So diffusion, passive transport, molecules will move in. And what if you have the same thing, right? You have the same thing exactly, but the cell, instead of taking in, the cell wants to take it out, right? This is against concentration gradient. Since it's against concentration gradient, diffusion is not going to work, right? Let me give you an example. There is like a club by the end of the day, right? Everybody left, and you have few people who don't want to leave. How are you going to make them leave?
I'm gonna start charging again.
How are you going to kick them out?
Security.
And security is going to do what?
I don't know.
Kick them out, probably.
Start pushing people out.
Pushing people out. To
They're
push
gonna
people
push it
out,
out.
what do you have to use to push people out?
Your hands? I don't know.
Force.
A taser.
Pressure.
You have to use energy. Come on. Okay, taser, electrical energy. Hands is a mechanical.
Yeah, that's a. I know with a taser, everybody's running.
But okay, the idea is you want to kick something out, somebody out, right? You have to use some energy. So this is active transport, right? Against concentration gradient. So you use energy. It's a molecule called ATP. It's an energy molecule. So cells are willing to spend energy to take substances against their concentration gradient. Does it make sense? Did I explain this well? Right? You have to take those who don't want to leave, right? So yeah, so for active transport, you have to you have to eat well, so you're gonna have this energy. And active transport is divided to primary and secondary. Do you have explanation? Not really much, but primary active transport, they have to use ATP directly. Last time, I'm gonna share the widescreen, sorry for me going back and forth. So for this, there's something called the pump. Active, good example is ATP pump. What is ATP pump? This pump will spend ATP molecule to take 3 sodium ions out and to bring 2 potassium ions in. The thing is that cells normally keep a lot of sodium outside and don't let it inside. And inside they keep a lot of potassium ions. So the pump pretty much moves both of them against their concentration gradient. You not necessarily have to pay attention to this right now. I'm not sure we're gonna return to this. But the thing is that that's how your nervous system works. Your nervous system, your neurons, the nerve cells, communicate to each other through generation of electrical current. To generate electrical current, they need these ions. They need sodium, they need potassium, and they need this concentration gradient of them. So your neurons spend 85% of ATPs running these pumps non-stop. So you can see a bunch of your energy actually goes to maintain your nervous system working. So these things might look weird and again, not really relevant to what you see, but actually they play a huge role in how your cells function. So yes, this is something that happens non-stop in your nervous system. Primary active transport,
My
ATP.
class right now, you heard? Ya se está terminando. Nah, I haven't seen him. Bueno, yo no sé, yo estoy clavado ahí adentro.
Steven, you can mute yourself when you talk to somebody, and plus I understand Spanish pretty well, so. And there is a secondary.
I'm sorry, Professor. I'm sorry.
Okay, secondary transport. Secondary transport, imagine you have one molecule that wants to move along concentration gradient. Let's say this is hydrogen ion. So hydrogen ion wants to enter the cell. And it can enter the cell just by concentration gradient. So sometimes energy of this one could be used to move something against its will, right? It's like when you enter the room to grab somebody and take out, okay? This is called secondary active transport. It's used through energy of another molecule, okay? But it's okay. I mean, I want you to read it. I want you to understand it, but that's, again, that's not the most important material, part of the material from today, okay? So it's not direct use of ATP, it's actually using energy of other molecules that move in and out of the cell. Okay, the last part about transport is something called endocytosis and exocytosis. So I told you cyto is cell, right? Endo inside, exo outside. Sometimes cells take big delivery, right? So big delivery, bunch of molecules that just, it doesn't make sense to let them in through diffusion or facilitated diffusion. Cell membrane is very flexible. It can actually make a pouch, right? Like a pocket, like this, loaded with their delivery and take it in. what you can see here, this bubble with the molecules, is called a vesicle, transportation vesicle, right? So cells take in big deliveries by just folding the membrane, right? Loading it into the vesicle and taking in. This is called endocytosis. Endo means inside. Engulfing large particles with plasma membrane. There are 3 types. Phagocytosis. Phago means solid, eating. Pinocytosis means liquid, drinking. And the third one is called receptor-mediated endocytosis. So let me just show you. Phago means solid, eat. In this picture, you see your white blood cell taking in germs. That's how your immune system works. It works through literally eating, feeding on germs, right? So your white blood cells, you have group of cells called phagocytes. Okay, and that's how it works. Swallowing, digesting, phagocytosis. If it's a fluid, this is phago. Fluid, like drinking, that's called pino. Pinocytosis is drinking fluid. Your cells don't do that, but some organisms that made of only one cell can do pinocytosis. Okay, and receptor mediated is like usually if there's a delivery, you see this cholesterol. So cholesterol is covered by like something like look like antennas, right? They have to bind to receptor, to sensors. So cell has to recognize first and then take in. So receptor mediated means it has to go through security, through clearance in order to enter the cell. So this is third type, receptor mediated endocytosis. Exocytosis is just opposite, taking contents outside the cell, right? So you load the vesicle will fuse with the cell membrane and release. So endo taken in, exo taking out. Is it clear? Okay,
Clear.
Again, terminology might look horrible, but on the bright side, guys, you don't have to memorize these words. You only have to recognize them. Your quiz is open book. As long as you know where to find the answers, and if words look familiar to you, you'll be fine. Okay, and let me see. Okay, we're almost done, actually. This one actually shows how cells connected to each other. If you talk about those that made of many cells, the organisms, if cell doesn't want to allow water to move in between, connection is called watertight seal junctions. Like basically, almost there is no room between the cells. What's going on, guys?
My hat fell by accident. I'm sorry.
What? What's going on?
I don't know. David said that his hat fell by accident.
Yeah, I don't know how that's funny,
Mute
David.
yourself if you're not talking so that you don't disrupt the class. Thank you.
Yeah, please, David. Like, come on, bro.
Come on, David. Okay, so this is tight junctions. Cells could be connected to like something like Velcro, right? That's called desmosins. Don't worry about these names. Cells could be connected through like tiny channels that run between cell membranes. That's called gap junctions, okay? So it basically shows that, again, cells can control how the membranes connect to each other. So, we're going to talk about the parts of the cell and we'll be done. Nucleus. So nucleus is their central piece inside the cell, the core. And inside the nucleus, you have genetic material, right? So it stores genetic material, and the DNA molecule, which carries gene, right, can look slightly different when the cell is dividing or when the cell is not dividing. When the cell is not dividing, it looks like bowl of spaghetti. Chromatin, right? So chromatin is basically DNA packed tightly inside the nucleus. This part is called nucleolus, which is involved in protein synthesis. As I said, nucleus has its own membrane. It's a double membrane, two membranes attached to each other, and there are openings between them called nuclear pores, okay? So bilayer means double membrane, and this is the pores for the molecules to come in and out the cell. Cell has its own internal frame. It's called cytoskeleton. Like we have skeleton made of bones, cells have skeleton made of proteins. There are three types of proteins that make up the cytoskeleton. The largest one called microtubules. They're like beams, like construction beams inside a cell. And they also allow certain things to move. So they work like roads, like highways inside a cell. There are proteins called intermediate filaments. Filament means like a fiber. So these fibers basically make cell flexible and strong. The tiniest called microfilaments, like ropes as well, they allow the cell to go through the division process. So one more time, cytoskeleton, internal skeleton of the cell. We have different types of bones. They have different types of proteins that make this up. Cells can have hair on the surface. They call cilia. Only sperm cells in mammals have tails. They call flagella. But bacteria usually have a lot of them. They have a lot of tails for movement. Okay, what are these for? Cilia can move fluids on the surface, like making like, how to say, this type of motion, wave-like motion, to move things on its surface. Flagella propels their cells themselves, okay? Mitochondria, I just explained to you. This is the organelle that is a power plant of the cell. And remember, mitochondria is the one that has its own DNA, right? So mitochondria is the one that takes oxygen and glucose and produces carbon dioxide, water, and energy. So you can say you breathe for your mitochondria. That's where the mitochondria goes to. Oh, sorry, that's where the oxygen goes to. The more active the cells, the more mitochondria they have. And what you can see, like, yes, you see, liver cells are very metabolically active, skeletal muscles, white blood cells, cells in the skin, right? So different levels of energy. But I don't know, guys. I mentioned already, working out. Have you ever heard that if you work out on the regular basis, you're gonna burn more energy between the sessions? Have you heard about that?
No.
Anyone here who used to go to gym on a regular basis?
Me.
Yes.
Yes, I did. I do. I still do go to the gym on a regular basis.
Yeah, David, stop it.
Multitasker.
So the thing is that if you train your muscles, what your muscles gonna do? Your muscles will respond by producing more mitochondria. The more mitochondria you have in the muscles, the more energy you burn. So it's true. So with the exercise, the key is doing it regularly, right? Because that's how your muscles change.
Professor,
Yes.
what does creatine and steroids do to the mitochondria?
They don't do anything to mitochondria. Creatine basically feeds your muscle cells with extra energy. Your muscles can take energy through oxygen, but they need mitochondria. If muscles running out of oxygen, they can use creatine as like a source of, it's like a backup resource. So nothing to do with that. It's just to keep your, keep your endurance longer. Steroids
Professor?
just help to make your muscles bigger. Yes, Xim. Yes.
I took pre-workout like 2 weeks ago for the first time, and I think I took too much because I was literally like shaking the whole workout at the gym.
What? You was what?
My whole body was like shaking. Yeah, I don't know, it's weird.
Yes. So it's like you start shaking pretty much because your muscles get super tired, right? They can't really support you. But the thing is that if you don't do it often, right? You do it once in a while, you get really sore, right?
It's not that much.
Yes, you
Yeah.
get, yeah. I remember like when I did it like after a long like gap. Next day I was just walking like holding the walls like to support myself because I was so sore. It happens because again, your muscles run out of energy very, very fast and they start turning to other sources. And when they start breaking down certain molecules for energy, they get a byproduct, which is called lactic acid. It's really acid. And that's why it burns next day, right? It hurts because of this acid accumulation. But if you do it again on a weekly basis, your muscles super dynamic. They get adjusted very, very fast. So that's not.
Oh, sorry.
Yes.
What
Yes.
about when you get, like, people sometimes get really sore from massages? Is it the same type of thing?
No, massage is something different. Massage, when the thing is that your muscles, all your muscles, also have nerves, right? Nerves that control your muscles. Sometimes massage, like when they try to, to break your muscles starts like they swell. When they swell, they put pressure over the nerve, and that's what causes pain. But not due to energy, or lactic acid. Okay, so I explained you about endosymbiosis. Lysosomes, organelles that actually do the sanitation, keeping your cells interior clean, right? So garbage removal. Okay, so as I said, protein synthesis takes place in this organelle called endoplasmic reticulum. It's a structure around the nucleus, and that's where ribosomes are, right? Ribosomes are literally protein building machines. They're located on endoplasmic reticulum, right? The rough is the one that has the ribosomes. Smooth doesn't have ribosomes, and other molecules are synthesized there. Fatty acids, phospholipids, steroids. Golgi apparatus is organelle that picks up newly made proteins, processes them, packs them, and ships them, either inside the cell or outside the cell. Yeah, you see a few slides left. Endomembrane system is present in eukaryotic cells only, right? So endomembrane system are organelles that can produce what's called the vesicles, the bubbles. Organelles that can make bubbles are plasma membrane, endoplasmic reticulum, both smooth and rough, and Golgi apparatus. And these vesicles, they move along the cell using cytoskeleton as a highway. So one more time, this is called transportation vesicle that could kind of break from certain organelles and move things around the cell. That's about animal cell. Plant cell has cell wall, which is made of cellulose. We talked about cellulose. And plant cells have like a bridges that allow water to move from one cell to another. These bridges go between the cells through the cell walls. They called plasma desmata, okay? It's like a channels that connect the cells. They have vacuoles. They store water, nutrients, could be used as a waste management. Predator deterrence, they can sometimes have like certain substances that repel pests, right? Could be used for reproduction, for physical support. That's vacuole. Chloroplasts used for protein synthesis. Or sorry, chloroplasts used for photosynthesis. Okay, so 3 organelles present in plants: cell wall, vacuole, and chloroplasts. This is a summary of organelles for you. Guys, as I said, this material might look tough, right? First of all, it's not. Okay, it's not if you just try to